Louis Pasteur. Louis Pasteur was born on 27 December 1822 he lived till 28 September 1895. He was a French chemist and a microbiologist renowned for his discoveries of the principles of vaccination, microbial fermentation, and pasteurization. So Louis Pasteur is revered by his successors in the life sciences as well as by the general public. In fact, his name provided the basis for a household word that is pasteurized. His research, which showed that microorganisms cause both fermentation and disease, supported the germ theory of disease at a time when its validity was still being questioned. In his ongoing quest for disease treatments, he created the first vaccines for fowl cholera, anthrax, a major livestock disease that in recent times has been used against humans in germ warfare, and the dreaded rabies. Fermentation and germ theory of diseases. Pasteur was motivated to investigate fermentation while working at Lilly. In 1856, a local wine manufacturer, M. Bigot, whose son was one of Pasteur's students, sought for his advice on the problems of making beetroot alcohol and sauce. So, according to his son-in-law, Remy Valradot, in August 1857, Pasteur sent a paper about lactic acid fermentation to the Société des Sciences de Lille. The paper was read 3 months later. A memo was subsequently published on 30 November 1857. In the memo, he developed his idea stating that I intend to establish that just as there is an alcoholic ferment, the yeast of beer, which is found everywhere that sugar is decomposed into alcohol and carbonic acid, so also there is a particular ferment, a lactic yeast, always present when sugar becomes lactic acid. Pasteur also wrote about alcoholic fermentation. It was published in full form in 1858. Johannes Jacob Berzelin and Justus von Liebig had proposed the theory that fermentation was caused by decomposition. Pasteur demonstrated that this theory was incorrect and that yeast was responsible for fermentation to produce alcohol from sugar. He also demonstrated that when a different microorganism contaminated the wine, lactic acid was produced, making the wine sour. In 1861, Pasteur observed that less sugar fermented per part of yeast when the yeast was exposed to air. The lower rate of fermentation aerobically became known as the Pasteur effect. Pasteur's research also showed that the growth of microorganisms was responsible for spoiling beverages such as beer, wine, and milk. With this established, he invented a process in which liquids such as milk were heated to a temperature between 60 and 100 degrees centigrade. This killed most bacteria and molds already present within them. Pasteur and Claude Bernard completed tests on blood and urine on 20th April 1862. Pasteur patented the process to fight the diseases of wine in 1865. The method became known as pasteurization and was soon applied to beer and milk. Beverage contamination led Pasteur to the idea that microorganisms infecting animals and humans cause disease. He proposed preventing the entry of microorganisms into the human body, leading Joseph Lister to develop antiseptic methods in surgery. In 1866, Pasteur published Etudes sur les vins about the diseases of wine, and he published Etudes sur la bière in 1876 concerning the diseases of beer. In the early 19th century, Augusto Bassi had shown that muscadinus was caused by a fungus that infected silkworms. Since 1853, two diseases called pebrine and flagellum had been infecting great numbers of silkworms in southern France, and by 1865, they were causing huge losses to farmers. In 1865, Pasteur went to Alice and worked for 5 years until 1870. Silkworms with the brine were covered in corpuscles. In the first 3 years, Pasteur thought that the corpuscles were a symptom of the disease. In 1870, he concluded that the corpuscles were the cause of pebrine. It is now known that the cause is a microsporidium. Pasteur also showed that the disease was hereditary. Pasteur developed a system to prevent pebrine. So after the female moths laid their eggs, the moths were turned into pulp. The pulp was examined with the microscope, and if corpuscles were observed, the eggs were destroyed. Pasteur concluded that bacteria cause flagellum. The primary cause is currently thought to be a virus. The spread of flagellum could be accidental or hereditary. Hygiene could be used to prevent accidental flagellum. Moths whose digestive cavities did not contain the microorganisms causing flagellum were used to lay eggs, preventing hereditary flagellum. Attenuating microbes for vaccines. fowl cholera, and anthrax. In the research campaign against disease, Pasteur first worked on expanding what was known about anthrax, but his attention was quickly drawn to fowl cholera. This investigation led to the discovery of how to make vaccines by attenuating or weakening the microbe involved. Pasteur usually refreshed the laboratory cultures he was studying. In this case, fowl cholera, every few days, he returned them to virulence by reintroducing them into laboratory chickens, with the resulting onslaught of disease and the birds' death. Months into the experiments, Pasteur let cultures of fowl cholera stand idle while he went on a vacation. When he returned and the same procedure was attempted, the chickens did not become diseased as before. Pasteur could easily have deduced that the culture was dead and could not be revived. But instead, he was inspired to inoculate the experimental chickens with a virulent culture. Amazingly, the chickens survived and did not become diseased. They were protected by a micro attenuated over time. Rabies and the beginning of the institute passed. Pasteur then wanted to move into the more difficult area of human disease, in which ethical concerns weighed more heavily. He looked for a disease that afflicts both animals and humans, so that most of his experiments could be done on animals, although here too he had strong reservations. Rabies, the disease he chose, had long terrified the populace, even though it was in fact quite rare in humans. After the time of Pasteur's vaccine, a common treatment for a bite by a rabid animal had been cauterization with a red hot iron in hopes of destroying the unknown cause of the disease, which almost always developed anyway after a typically long incubation period. After developing the rabies vaccine, Pasteur proposed an institute for the vaccine. In 1887, fundraising for the Pasteur Institute began, while donations from many countries. The official statute was registered in 1887, stating that the institute's purpose were the treatment of rabies according to the method developed by M. Pasteur and the study of virulent and contagious diseases. The institute was inaugurated on 14 November 1888. He brought together scientists with various specialties. The first five departments were directed by two graduates of the École Normale Supérieure, Émilie Ducalax, that is General Microbiology Research, and Charles Chamberlain, that's Microbe Research applied to hygiene, as well as to a biologist, Eli Metchnikoff, morphological microbe research, and two physicians, Jacques Joseph Greiner, rabies, and Emily Roque's technical microbe research. 1 year after the inauguration of the institute, Roques set up the first course of microscope microbiology ever taught in the world, then entitled Course de Microbe Technique, that is, Course of Microbe Research Techniques. Since 1891, the Pasteur Institute has been extended to different countries, and currently there are 32 institutes in 29 countries in various parts of the world. Regarding his death, in 1868, Pasteur suffered a severe brain stroke that paralyzed the left side of his body, but he recovered. A stroke or uremia in 1894 severely impaired his health. Failing to fully recover, he died on 28 September 1895 near Paris. He was given a state funeral and was buried in the Cathedral of Notre Dame, but his remains were reinterred in the Pasteur Institute of Paris in a vault covered in depictions of his accomplishments in Byzantine mosaics.